I'm Abdulazim from Group 4. So today we're going to present about continuous measurement. Hello, Iman, can you share the screen? Hello, okay. Today we're going to present about continuous measurement of cholesterol. Okay, so first we go to the next slide, which is what is cholesterol? Okay, what is cholesterol? Cholesterol is an organic molecule from a plant or animal. It is a type of lipid, which is fat, in an easier way or easier language to tell others. It is a fat that can be found in the blood. It is a waxy substance, can be accumulated or can be built in our arteries. So there are two types of cholesterol, which is saturated and unsaturated. For saturated is a low density of lipoprotein, which is LDL, and it is can described as a bad cholesterol. LDL is a cholesterol. It can contribute to fatty build-ups in the arteries, and then LDL or saturated cholesterol is made up from a single covalent bond between carbon atom. Okay, for unsaturated or high density lipoprotein, which is as known as HDL, it is known as a good cholesterol, which is it carries excess cholesterol in the blood to the liver, and it will broken down or digest the bad cholesterol or LDL, which is saturated, to digest and be removed out of body. And it is made up from it's made up with double covalent bond. And then we move to the next slide, which is what is the function of cholesterol. We have three main function cholesterol to the body. It's first to maintain the integrity and fluidity of the cell membranes, which is it can maintain the shape or maintain the cell membranes or the wall of the cells. Then the next one is it helps liver to produce bile, or in Malay we call empedu. And then the last one is reduce permeability of the cell membranes. As I said before, it helps to 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 enhance the cell membranes. Okay, so we move to the next slide, which is the importance.
Hello. So the importance of cholesterol measurement. So the continuous measurement can help to monitor the cholesterol level in blood. So for early detection of any disease or risk, so that we can go to the treatment. Okay, and then the imbalance of cholesterol level can cause disease and affect body performance. So the balance of the blood cholesterol between HDL and LDL is very important because the imbalance usually can cause disease and affect the body reaction. So there are several studies that relate human behavior and psychopathology with cholesterol. So the study relate between the cognitive function of a human with their cholesterol levels. So some of the human behavior is depression, suicide, personality and aggression, depression, attack, personality traits problem, and violent behavior. So the study suggests that there is a correlation between cholesterol level and the human behavior, but there is no the evidence behind this is actually still unclear. So the common problem that with relate with blood cholesterol is actually the high LDL, which referred to as bad cholesterol, as Azim stated, in blood can cause formation of plaque in arteries, which can lead to heart attack, coronary heart disease, and stroke. So as you can see the small picture in the right below, so when there are too much concentration of LDL in blood artery, it can cause a blockage, which can slow the blood flow, which can reduce the oxygen because blood carry oxygen to certain part of the body. So if the uh, oxygen level is reduced in the brain, for example, can lead to stroke. And then if blood pressure is low to the heart, which can cause heart attack and other heart related disease. So this is the importance of cholesterol measurement. So next, about the mechanism.
Hello. Now I will be explaining a real-time, non-invasive cholesterol monitoring system. Next slide. So this technique is actually a non-invasive for monitoring blood cholesterol levels using sensors that utilizes infrared light absorption in body tissues. It is actually to simplify the measuring blood cholesterol levels regularly for the patients. And it is focused on developing a total blood cholesterol monitoring device using an infrared sensor with infrared LED 940 nanometer wavelength as a transmitter. Next. So the measurement of blood cholesterol levels with this technique, the non-invasive technique, it is done by conducting blood irrigation on body tissues, such as the earlobe and fingers, using the wavelength. So the light absorption at wavelengths is utilized by measuring transmission or reflectings. So the use of this technique includes the infrared as a spectroscopy technique that employ the infrared wavelength region of an electromagnetic spectrum. Next. So in this device, there is a photodiode that is used as a detector with wavelength range of 400 to 1100 nanometer, and also a microcontroller as the minimum system for controlling the value of the output voltage in the form of digital data, and then converted into total blood cholesterol. Next. So determining blood cholesterol concentration is related to the light reflectance angle. It has a direct correlation between light intensity and output voltage. So to measure the cholesterol levels, here, if you can see in the figure, the LED transmitter and a photo transistor, it has lines and points. You can see A, C, and B, and angles as well. So to measure the cholesterol levels, it's by connecting points A, B, and C. So the increase of cholesterol levels in blood can reduce the reflection angle, as you can see, angle 2, so that the line of AB moves closer to the line AC and creates the new line AB. So mathematically, the refractive index can be calculated using Snell's law. So the reflection angle angle 2 decreases when the blood cholesterol level increases, which reduces the reflected index and increases output voltage because of many photons touch the photodiode. Next. So as a result here, as a result of the decrease in the reflection angle, the radius of point BC in LED light also decreases when the light rays curving to AC, which causes the increase of cholesterol levels according to Snell's law. Next, Afif will be explaining the mechanism of the device.
So for the methodology process is starting by initialization. Is convert the analog to digital by processing the signal from the infrared LED sensor. The LED, the sensor will pressing by fingertip. So the issue will be absorbed by the stream, and the others cannot be absorbed. It will be reflected and accepted by the photodiode. So the voltage from the photodiode will be supplied to the ADC microcontroller, and it will be calculated by using algorithm equation and the total of cholesterol and the reading can be displayed on the LCD and also can be sent to the smartphone user for the guide. For the next slide, for this slide, the figure shows the series of the sensor consists of IR LED. So this IR LED emitted to the infrared light, and for the photodiode is used for the detector. So it will be absorbed and receive the light. So when the sensor was installed at the fingertip, the light from the LED will be absorbed via tissue in the fingertip, and the others will be by the receive by the photodiode detector. After that, the light will be received by a photodiode to become a light value. So it will convert to electrical current. Then it will become the voltage to present of the light of the resistor. We can see in the circuit diagram. The voltage will be released by photodiode is depend on the amount of the light received. So the more the light received, the greater the voltage will be released by photodiode. It will be arranged for 0 to 5 volts. For the voltage value of the photodiode will be still a low, so the variation in the voltage value is less, will be noticeable. So to deal with this, the IC amplifier circuit will be installed to make a strength the voltage from the sensor. The voltage value of the photodiode from the sensor will be read by ADC microcontroller, and the ADC value it will convert into the voltage value. Then it will be enter into the linear equation to change the total blood cholesterol value, and the data from the total blood cholesterol will be convert and we will measure the result will be displayed on LCD and also smartphone. Next slide, I will pass to Izzat to explain about the strength and weakness.
For the strength and the weakness, from the study, we focus on the strength and weakness of several elements of invasive vs non-invasive techniques to measure the level of the cholesterol. Basically, we focus on developing tool blood cholesterol using infrared sensor or infrared light, and propose and present the idea measuring cholesterol level without need of the blood sample compared to the existing invasive method that if they want to measure the blood cholesterol, they need to take the blood sample. For the strength, the first point, the non-invasive techniques measure the parameter without the blood sample. So basically, this method system is more comfortable for user and patient in order to continuous monitoring patient, such as especially the child that admitted in the hospital, in the ward, and in the ICU. For the second point, the non-invasive techniques are easy to use. The result of this developmental alternative innovation, more efficient, low cost, less expensive than existing invasive method, and not painful because it does not use cholesterol strip. And third, non-invasive technique accuracy result has developed quite well and reliable, same as invasive technique. Non-invasive technique, it use infrared light, and invasive technique, it need to take the procedure such as take the blood sample. So from this, the user and the patient can get the good feedback and the good recommendation. Either they want to use the non-invasive techniques or invasive techniques. Okay, so for the weakness, the first point, as we know, non-invasive technique or non-invasive model, they are fully relies on the infrared sensor, infrared light or NIR sensor detection. So the result, the accuracy of the result relies and depends on the performance of the sensor detection. Second, for the invasive techniques, the procedure include the collection of the blood sample, which pose or expose the patient or the risks such as inflammation and bruising. And lastly, non-invasive technique sensor detection are very sensitive, and it may causes from the not powerful enough to clamp a patient finger. Second, risk of light entering and being detected by photodiode, and the result of the thickness of the patient finger also may affect the accuracy of the level cholesterol result. So I will pass to Arifa for the feature view.
For the challenges for this device, many challenges had been faced throughout the progress of the device. The developer faced problem displaying many details on the LCD. There are also difficulties to display all input and output values on monitor because at the first, haven't installed some libraries in device and lacking knowledge to handling the software. After fix all problem, they successful make the device worked very well. Then for chance commercialize, is that yeah. Is that sorry, yeah, for the chance commercialize, based on our analysis, the device have a chance to commercialize because use a IR LED and photodiode to detect the cholesterol level. It is more efficient than the invasive method and non-invasive measurement. The result can be displayed on LCD and smartphone as the output because have mini wireless data transmission module inside. The non-invasive method is fast measurement and accurate. Also, the system is user-friendly that everyone can use it. I think that's all from my group. And thank you.
Okay, thank you for the presentation on question one. Any questions? This is now open for the Q&A. Do you have any questions?
Hello, Dr. Hello.
Okay, yes, yeah. You may deliver your questions.
My question is, let's in the problem, they stated that too many details in the device, too many details cause the confusion something. Then how what kind of details they make the patient confused, and how it's then they mention about user friendly. So how they wanna overcome these two problems? Is it clear?
Okay, now the previous slide. Please share the presentation slide. Group 4.
Thank you.
Hello? Yes. No, previous slide. Yeah, problem display many detail on LCD. I believe that the question is asking why display many detail and why it is. Is it?
Yeah, yes, doctor.
Yeah. So group 4. So what do you mean by displaying many details? And then what is the causes of this thing? And then why it is stated as a problem?
Doctor, because at the first, when the developer making the device, they face a problem, the difficulties to display all input and output to monitor because lacking of knowledge.
So the question now, so the problem is mean current device only show limited detail, but the thing is you want to show a lot of detail it is not easy. Hello, group 4.
Yes, doctor. Sorry again.
No, I mean, so the current you mentioned here, problem displaying many detail on LCD. Is it the problem to show many detail is a challenge, or is it showing too many detail is a challenge?
No, the challenge is the LCD has limited display, so they want to display more.
You mean LCD has a limited port to display more?
No, the size of the size of LCD when developed can't really display many apa ni not actually many details when they develop. So the challenge is they want to display many details, so they need to upgrade the LCD or something in the future because this is a future view.
It's okay. Actually, I have a lot of questions, but I cannot ask. If not, then you cannot answer. So, is it answering your question?
Yes, yes. Not clear, but yes, okay. Somehow.
Okay. If you say so, then okay. If you're satisfied. Okay. Any more questions? Let's count to 5. 5, 4, 3, 2, 1. Okay, thank you so much for the group 4, section 1.